as the head position changes. Okay? Yeah?
I'm trying to find this material in the book, and I'm having a hard time.
You need the chapter on special senses?
Okay. Chapter 15.
Okay, special senses, there you are.
Oh, I just, look at that.
Bottom page.
All right.
Okay,
Thanks.
all right. The last one of these sensor tissues or sensor apparatus is the one which is related to dynamic movements of the body, the semicircular canals. Okay? There are 3 of these. You'll see these in the laboratory. Okay? And they are oriented in different planes. In the lab, you're likely to see one of these semicircular canals sort of standing up in the coronal plane. Now remember, these things are on both sides of your head. And another one is going to be in like the transverse plane. And then another one is in kind of more like a sagittal plane. And so what you've got then is on both sides of the head, you've got the ability to detect movement in multiple directions. Either way, so even if you're spinning around and your head is down like this, you can still detect the spin. You don't have to be standing straight up. Okay? All right. Membranous semicircular ducts line each canal and communicate with the utricle. Okay, then you can just set that aside. What's more important is at the base of these membranous canals is another sensor tissue known now as ampulla. No longer macula, now ampulla. Okay? You're going to see a diagram of this. And in the ampulla is a tissue that looks a lot like a macula, but is a little bit different. It's called the crista ampullaris. Okay, this is the crista ampullaris. This is the ampulla, okay? And then this is a gelatinous material known as the cupola. Take a look down here as best as you can. You are seeing hair cells and supporting cells. You are seeing that they are synapsed with neurons. You are seeing that they have hair cells, or excuse me, stereocilia, and that the tallest in the bunch is called the kinocilium. And if you looked up really close, you would find out that the kinocilium is stuck in the gelatinous material known as the cupola. So try to think about this thing having fluid in it, and as long as the body is static, the fluid is at rest. Okay? The fluid's at rest. But now say, if I spin my head that way, the fluid eventually begins to move in these circular tubes, right? Okay? So this thing is extended into a circle, as you can see. So there's fluid in there, and as long as we're static, the fluid just sits. And the cupola sits straight up, and there's a low frequency of ax potentials leaving the crista ampullaris. But the minute we spin the head, the fluid begins to move, and it leads to deformation of the cupola, just like the movement of the otolithic membrane. Now, what's causing the gelatinous material to move is not gravitational effects. It is rotational movements. Okay? What you'll read about this, if you're very careful with your reading, is that eventually, as fluid begins to move, it eventually stops moving, even though the head may still be spinning around. Okay? You have to read this to understand a little bit better. But as I start to spin my head around, as I begin the spin, the fluid tilts. But eventually, as I keep spinning, the fluid becomes static again. Okay? Until I stop spinning, and then the fluid tilts again. So what this system does is detect the beginning of rotation and then the end of rotation. Okay? And it will make adjustments as a result of the beginning of rotation and the end of rotation. What I've read about this all of us have gotten dizzy at some point, purposely, by rotating around. Okay, it's really fun to watch when adults play children in baseball and the adult has to spin around a baseball bat. It's really fun to watch that, to see what happens afterwards. Okay? And in reading your book, they talk about how once the spinning is over, how people have difficulty maintaining balance. The brain has detected the spin and has made adjustments to try and maintain posture and eye movement during the spin. When the spin ends, it takes a little while for the brain to put things back. And so the brain is still compensating for the spin, even though you're not spinning anymore. And that is true in eyes as well. You may, if you get somebody really dizzy, when you stop it, you may see their eyes still flitting as they are trying to keep up with the image moving in rotation. You follow me on this? Okay? All right? So it really is about then connecting to postural muscles and to the eyes to get them to compensate. Just like the macula, depending upon which way the body moves or the head rotates, is going to then alter the action potential frequency from the crista ampularis. As you can see here, the saying flow of endolymph in one direction, okay, is indicating to the brain rotation in one direction, okay? And then when it flows in the other direction, it's indicating to the brain that the rotation is stopped or you are rotating in the other direction. It all comes down to then the frequency of action potentials coming from the crista ampularis. Low frequency action potentials tells the brain that you're rotating one way. High frequency action potentials tell the brain you're rotating another way. And a steady rate of action potentials tell the brain that you're not rotating at all. Okay. The micrograph does a nice job of showing us just kind of what that thing looks like there. Just a little gelatinous mass, which is affected by fluid. Remember that the fluid in here, the endolymph, is a high potassium fluid. So as we bend kinocilia, we open tip link, or we open our mechanical gates, and we allow potassium to come in and we depolarize. If we move in the other direction, we close those gates, and the cell hyperpolarizes. All right. Do you have any questions then on the special senses? We looked at the eye, but we didn't look at how to, let's say, fix the eye if it's not able to see things in focus. But your book does talk about that. We didn't talk about color vision all that much, and how there's a different set of opsin proteins that are used, and you could read about that if you choose. Sarah, did you have a question or something you want to say?
No.
Nothing at all? Okay. Yes?
So, with the ear, basically everything's off of sound waves and fluid movement.
Yeah, and pressure. Yeah, pressure waves. Yes, absolutely. And it's all mechanically gated ion channels. Okay? And in the eye, then, it's still using, your ligand-gated ion channel, but it's sort of the opposite of what we expect. If we had done olfaction and taste, we would see even other ways in which receptors are used. Okay. Well, then, the last chapter we cover is